Hello everyone, my name is Angela and along with my group we are going to present our project named Targeted Mutation Calculator. Our project is based on recombinant protein expression in bacteria. We aim to design a program that can take in DNA sequences from many bacteria and amino acids and a desired protein and output the most suitable bacteria based on the least number of required mutations to produce specific protein. For our workflow diagram, we start off with two files, the first one being the desired protein which is generated by the user as a text file, and the second file is the amino acid database. We have a Python script that uses these files to generate a new file containing all of the possible sequences that encode for the desired protein. Then this file, along with the bacterial sequence database, would be used to compare each bacterial sequence to all of the possible sequences encoding for the desired protein and pick out the sequence with the least number of mismatches. Then, if the protein sequence is the closest to the bacterial sequence, the protein is saved as this closest sequence, and if not, the algorithm will keep on looking. The algorithm will proceed to make a general report which contains each bacteria, the protein sequence that is closest to it, and the number of mismatches, and then prints out the bacteria that has the least number of mutations. The user will decide if they would like to download a more detailed report for that specific bacteria that compares each bacterial sequences to the protein sequence and the mutations required.
Now let's take a closer look at the code. The first helper function, makeLonger, takes two parameters. The first is a sequence S as a string, and the second is an integer L, which represents how much longer you want the sequence to be. In this case, we're assuming the sequences we would like to make longer have a trailing gap, so this function adds L number of dashes to the end of the sequence S and returns the modified sequence. Next, we have compareSeq. This function takes two sequences as strings and returns the number of mismatches between them. It starts by checking if the sequences have the same length. If not, it calls the above helper function makeLonger to make the shorter sequence as long as the longer by adding dashes to the end of it. It calculates the length difference between the sequences and passes that into makeLonger function along with the short sequence. Then we convert both string sequences to uppercase letters since the next step is comparing them. To compare, we iterate through the base pairs of both sequences and check if they're equal. If not, it adds to the count of mismatches. In the case where a sequence is shorter, it will have dashes at the end of it as the trailing space, and each gap will be counted as a mismatch. The function then returns the total number of mismatches between the two sequences. Next, we have bacteria recommendation. It takes in the general report as a pandas data frame and prints out the recommended bacteria, which corresponds to the bacteria with the least number of mutations needed. So the general report has three columns. The first is the bacteria, second is the protein sequence, and third is the number of mutations needed to go from bacteria to desired protein. So what this function does is it finds the minimum value of the mutations column, then goes through the rows and prints out the name of the bacteria or bacterium with that corresponding number of mutations. Finally, we have detailed report. This function takes in the bacteria's ID and the general report and creates a new detailed report for that specified bacteria. It starts by getting the data about that specified bacteria from the general report, so the bacterial sequence as well as its designated protein sequence. It then makes sure that these sequences have the same length and compares them by iterating through the nucleotides of the sequences and writes the mutations that need to be done to go to the bacterial sequence in a text file that is then saved into the working directory.
After looking at the helper functions, we can go ahead and run the program. After running the program, the user can display more information, run the program, or exit. If the user types info, it will display general information about the function of the program and what will be outputted to the user. It will also display information about the required formatting of the input file of the desired amino acid. If the user types run, the program will run and ask the user for the name of the file containing the desired amino acid sequence. In order to successfully run the program, it will take in the amino acid database, which needs to be used as is from the GitHub, where each column is the amino acid and beneath them are all the corresponding codons, and save it to a data frame. It will also take in the name of the file with the desired protein sequence that the user inputted, where each amino acid is in a line in order from start to finish, top to bottom, and save the amino acids in the sequence into a list. Using the function product from itertools, it will take in these inputs, where each input corresponds to all the possible codons for one of the amino acids in the sequence. The possible DNA sequences will be saved to a CSV file that will later be used by the bacteria recommendation function. The user will need to know how many amino acids are in their sequence and need to update the inputs into this function accordingly. After the file name is inputted, the program will output the bacteria recommendations, which are the bacteria containing the least number of mutations using the function bacteria recommendations. In the future, we hope to further refine our algorithm to provide a smaller list of recommended bacteria to the user. The program will also read in a CSV file containing the bacteria that the user wants to compare to their protein, which includes the bacterial ID, the corresponding sequence. After that, the program will iterate through all the possible sequences for the desired protein and compare it to the bacteria sequences with the compare-seq function and output the bacteria recommendations using the bacteria recommendation function. It will also save the bacteria ID, sequence, closest protein sequence, and the number of mutations to a report that is saved as a CSV file. After that, it will ask the user if they would like to download a detailed report. If the user chooses to download a report, it will prompt the user to input the name of the bacteria they want the report for. It will then call the function detailed report, and it will save the report as a text file and exit the program. The detailed report has the bacterial ID, bacterial sequence, protein sequence, and a step-by-step list on how to convert the bacterial sequence in the desired protein sequence. If the user does not want a detailed report, it will exit the program.
So here is our user interface code. So I'm just going to run the code here. So when you run the code, you have the program's name, you have a short description of the program, and then you also have a little note that ensures that the user is using text files. We have two options, info and run. We're just going to go through the run portion here. So when the user clicks run, they have the option to enter in the amino acid file name here. Afterwards, if they want to download a detail-based report, they can click yes or no. And then afterwards, again, they have to make sure they select the type of bacteria that they want to use, which is from bacteria A through Z. Afterwards, they generate the program. So we have here, this is the homepage code. This is more purposely on aesthetic purposes. The main part of this code is this portion, which is going to call out the BE552 final report start. So when the user clicks the start button, it would run this code, which again allows the user to enter in the amino acid name, which will call in the file from their respective laptop or computer. Afterwards, again, they have the drop-down menu where they select the bacteria A through Z, and again, the rest of the code on the bottom is again for aesthetic purposes. In the future, we definitely would like for this program from go to MATLAB to Python so it's more accessible to the previously code you've seen previously in the video.